everyone. In today's module, we'll be talking about C3 versus C4 photosynthesis. First, we'll define what exactly is C3 and C4 photosynthesis. Then we'll talk about what makes C4 plants so special, in particular, and how they overcome photorespiration. And then we'll finish with what environments favor C3 and C4 plants. So we've been talking quite a bit about the Calvin cycle and what happens with rubisco in different environmental scenarios. Now, what I haven't mentioned yet is that this entire process that we've covered so far is known as C3 photosynthesis, and the plants that use this mechanism of photosynthesis are called C3 plants. So why are they called C3 plants? Well, it's because the first product after CO2 gets fixed is in fact a 3-carbon compound. So that means the first product here is a 3PG. As we know, we have another one. So there you have it. That's why it is called C3 photosynthesis, and it's because the main product after we fix the CO2 is a 3-carbon compound. And of course, we know that C3 photosynthesis is very susceptible to photorespiration. And of course, there are ways to work around this. And so in today's module, we'll be talking about some cellular arrangements that other types of plants have utilized in order to deal with this photorespiration issue. So let's take a step back for a moment and revisit this overall anatomy that we see within a typical leaf. So in the book goes through this diagram and this arrangement, and this is for a typical C3 plant. And we've covered the general features here that yes, we have all of this between the upper and the lower epidermis, just some mesophyll cells. As you can see, they are green, they have chloroplasts, and the cells, these mesophyll cells toward the upper epidermis tend to be more tightly packed, so we're trying to maximize capturing photons of light. And we also have these intercellular air spaces here. And as we know it from talking about before, during the vascular tissue system section, that we have tightly packed xylem and tightly packed phloem here together that comprise the veins and the vascular tissues that we see throughout the leaf. And I really want to draw your attention to what is surrounding those tightly packed xylem and phloem. And so here you can see these individual cells all nice and tightly packed and surrounding and bunching those xylem and phloem together. So these are the bundle sheath cells. So this in general is what we typically see in the arrangement of cells for C3 plants. So what about plants that are different from this? So if we think about this typical cellular arrangement that we find for C3 plants, well, C4 plants for the most part have a very similar arrangement. There is a key difference, though, in that in addition to the tightly packed bundle sheath cells here, we also have another ring of tightly packed mesophyll cells exterior to the bundle sheath layer. So we in fact end up with this kind of double ring structure of tightly packed cells. On the exterior, it's the more mesophyll cells, and on the interior, this typical bundle sheath cell layer that we see. And this is known as the Kranz anatomy. It's a very classic anatomy term here for plant physiology. It's German for wreath, so it honors the wreath-like arrangement of mesophyll cells around the bundle sheath cells. And the plants that have this type of anatomy are known as the C4 plants. So let's talk about why this cellular arrangement is so special for C4 plants. So of course, here's another diagram. On the interior, again, we have the vascular tissue that contains the xylem and the phloem. Here is our ring of the bundle sheath cells, and this outer ring that is the tightly bound mesophyll cells. And each group of cells has a very specific enzyme contained inside them. So for the bundle sheath cells, they only have the enzyme rubisco, and the mesophyll cells on the exterior only contain a new enzyme, a new key player here called PEP carboxylase. PEP stands for phosphenylpyruvate. So again, another good abbreviation of just PEP. And in looking at the full name of this new enzyme, PEP carboxylase, so it should sound slightly familiar, as in it's a carboxylase, so that means it's going to fix CO2. There is a strong interconnectedness between the mesophyll cells and the bundle sheath cells. So let's zoom in a bit to understand what is happening here. And if we zoom into the mesophyll cells, again, this is the exterior ring of cells. This PEP carboxylase right here, that is our enzyme that's going to kick off CO2 fixation. And again, this CO2 molecule right here, that has diffused inside the leaf from the atmosphere. So it's worked its way through to this cell where it meets up with PEP carboxylase. So PEP is the enzyme that's going to help merge CO2 with a CO2 acceptor molecule. In this case, it's PEP, phosphenylpyruvate, instead of what we've learned before, which is RUBP. So PEP is going to merge with a CO2 molecule with the help of PEP carboxylase. And the first main product of fixing this CO2 is a C4 compound here. So that is where these plants get their name from. The first product is a C4 compound, therefore they are the C4 plants. And all of this right here with the PEP and the PEP carboxylase and the CO2 and the C4 compound, this is only happening in the mesophyll cells. It's also really important to note that PEP carboxylase is very different from Rubisco in that it does not have an affinity for oxygen. So I guess the key feature here is that PEP carboxylase will always want to fix CO2. Therefore, in this very first step, it is super efficient, way more efficient than Rubisco could be, because Rubisco might want to also fix oxygen. Well, here we've eliminated that, and PEP carboxylase only wants to fix CO2. And at this point, Rubisco hasn't really done anything because the first step involved PEP carboxylase, and we formed this C4 compound here. Now, I'm not going to get into the nitty-gritty details again of all of the reactions that can possibly take place here. Just know that there are a series of reactions where we get some intermediate products from the C4 compound. These then get transported into the bundle sheath cells. And here we get this really cool split. So the intermediate compounds, they will split into a CO2 molecule that starts saturating or building up in concentration in the bundle sheath cells. And we also get this 3-carbon compound that will simply go back into the C4 cycle, all to regenerate PEP. But returning to the bundle sheath cells here, again, after we have these intermediates that have come in, it splits and it gives us a CO2 molecule. And this helps to build up the CO2 concentration only in the bundle sheath cells. And then finally, Rubisco can get to work. And in this particular case, Rubisco has no choice but to act like a carboxylase. So it's only going to fix CO2 because we've created this high CO2 environment within these bundle sheath cells. And Rubisco and RUBP and CO2, they will all merge and do their regular cycle in the Calvin cycle here. And the Calvin cycle is only taking place in the bundle sheath cells. So we get this spatial separation of the enzymes, and that's really important. And so in the mesophyll cells, again, we only get PEP carboxylase, only fixing CO2, and through the process of getting the intermediates and getting more CO2 that is built up in saturating levels inside the bundle sheath cells, that only allows Rubisco to act as a carboxylase and only fix CO2. Thus, we eliminate or strongly reduce the photorespiration that we typically see in C3 plants. So C4 plants, even though we might have a little bit of extra energy used up in the form of ATP during this process, and we have another enzyme, PEP carboxylase, this is way more efficient than what we've seen previously with plants that only use Rubisco. So this is a really fantastic fix for the photorespiration issue, right? C4 plants have found this workaround, even though they might have to use up a little bit of extra ATP and have an extra enzyme on hand. This is a really great way to reduce photorespiration. And some of our most important crops are C4 plants like corn and sugarcane. These are produced in such high amounts for our daily life. And so that is really fantastic that these plants have utilized the C4 mechanism. Now, I guess the problem is that nearly 90% of the plant species that we see are C3 plants, and that represents a huge portion of the Earth's total plant biomass. So we may see this really great mechanism of dealing with photorespiration in C4 plants, but we're only going to find that in this particular group, and as well as only in particular areas of the world. So in hot and arid climates, we tend to see that C4 plants are doing really well, or we might see them in higher abundance there. So it'll be really interesting to see what happens in the coming decades. How will C3 plants and C4 plants transition their distribution across the world given a change in climate? So let's think about that in an exercise here. So we know that we are seeing an ever-increasing atmospheric CO2 concentration. So that's on the y-axis here, atmospheric CO2 concentration, from 100 all the way up to something really high. Just as a frame of reference, we have surpassed the 400 mark. So on average, the world is seeing a daily average of 400 or 410 in terms of our average daily atmospheric CO2 levels. And of course, this will only continue to increase in the coming decades. And concurrently, we're seeing an increase in daytime growing season temperature. Again, that's on the x-axis here. So this is a graph simply representing the different possible combinations of growing season temperature and atmospheric CO2 concentration. And this is a very classic graph. It's been around for quite a while, but it lets us think about, well, who's going to win out? What type of plant is going to do really well in certain combinations of growing season temperature and atmospheric CO2 levels? And as a frame of reference, this red line here, this is indicating that C3 plants and C4 plants are equivalent in their ability to do well or have great productivity given certain combinations. So in this case down here, if we have daytime growing season temperature of around 10 and atmospheric CO2 concentration of 200 or a little bit less than 200, then C3 plants and C4 plants will both have about the same level of productivity. And of course, something more realistic to where we are atmospheric wise is around 400 right now. And we will only tend to see an increase in this daytime growing season temperature as time goes on. But this reference line is they are both equally good at that particular combination. So now I'd like to ask you to think about who's going to win out if we get above this reference line. So anything above in this area here, kind of the top left corner of this graph, who is going to win out and do better? Also, let's think about at the bottom right part of the graph, below that reference line, who is going to do better, C3 plants or C4 plants? So there's kind of a lot to unravel here, or a lot to think about to rationalize who will do better in either one of these scenarios. So take a minute to press pause here and think about it, and then we'll get back to it in a second. So after you've finished thinking about who's going to win out in certain combinations, on the top left or above that reference line, C3 plants will be favored, and this is because of the photorespiration issue. So if CO2 concentration is really high, then more of it will be able to diffuse inside the leaf, making Rubisco act more like a carboxylase. Now C4 plants are a little bit different here, again, because of the photorespiration issue, or more specifically because they have found a workaround for it. C4 plants are already really efficient at concentrating their CO2 inside the bundle sheath cells. So they don't really benefit, C4 plants don't benefit from that extra CO2 because they are already really efficient and already have found a way to concentrate CO2 inside their cells. C3 plants obviously have not found a way to do that, and with an increase in CO2 levels, they respond to that really, really well. So that kind of adds an interesting conundrum here. We don't really want an increase in atmospheric CO2 levels, but C3 plants will tend to respond very positively to it and be more productive. Conversely, if we look at the bottom right part of the graph, anything below this red reference line, C4 plants are favored. And again, this gets back to that photorespiration issue, meaning that if it gets really, really hot, really high temperatures, as we are expected to see in the future, that the stomatal pores will tend to close in order to conserve water loss. So C4 plants, because they have this concentrating mechanism of getting more CO2 inside the bundle sheath cells, they can actually be okay with that. Even if the stomatal pores were getting really, really close to closing, that they still have high CO2 in their bundle sheath cells, and they therefore will do really well in a high temperature environment. And at the same time, C3 plants are very, very sensitive to these rises that we see in temperature. So they won't be favored here. So it becomes this interesting back and forth. Who could do better given an increase in temperature versus an increase in atmospheric CO2 levels? So, but I guess if I were to think about where are we today, we are at 410 or so, and our daily temperatures are only going to get hotter. I'm going to say in the long term, C4 plants are going to be much better off. However, they represent a small portion of what we see out in the environment. And for another exercise, let's compare the CO2 response curves for C3 versus C4 plants. So I will start by drawing a graph that I think we could possibly see for a C3 plant. And again, this should look very familiar to what we covered in Module 2 in thinking about CO2 response curves. That was for a C3 plant. So now what I want you to do is think about how might the graph be different for a C4 plant. Now we've talked about photorespiration and the different cell arrangements involved and how C4 plants have dealt with the photorespiration. So let's compare them now. So take a moment to press pause and draw out a graph that represents the CO2 response curve for a C4 plant. Now that you're back, let's go over the differences between the CO2 response curves. For a C4 plant, because it has this great ability to concentrate CO2 in the bundle sheath cells, it makes Rubisco much more efficient. And therefore, this first part of the graph should be incredibly steep. So that linear portion of the graph, way more efficient than what we see for a C3 plant. So that happens. But I guess the trade-off here is that because it's already really efficient, it's not going to benefit that much more from increases in CO2 concentration. So it'll start to level off, and it might not achieve as high of maximum photosynthetic rate because it's already really great at concentrating the CO2 levels in the bundle sheath cells. Another thing to point out would be the CO2 compensation points. Again, this refers back to this dashed horizontal line at 0. Again, this is the net carbon assimilation rate at 0. That means that respiration and photosynthesis balance each other out. And because C4 plants are much more efficient with increases in CO2 levels, they achieve that CO2 compensation point much earlier than we see for C3 plants. So this graph summarizes the main differences between C3 plants and C4 plants, all because of this photorespiration issue. So the main takeaway from this would be that C4 plants are incredibly efficient at fixing CO2, even at low CO2 levels inside the leaf because of that concentration mechanism inside the bundle sheath cells. On the other hand, C3 plants in the long term are going to respond and be sensitive to rises in atmospheric CO2 levels. So again, lots to think about as we look to the future and how changing climates might affect productivity of plants. That's it for today's module. Your main takeaway should be first, how C3 and C4 plants have very different cell arrangements, and how C4 plants use 2 enzymes instead of 1, and that these enzymes are separated spatially between mesophyll cells and the bundle sheath cells. Your second takeaway should be that C4 plants reduce photorespiration greatly and are very efficient relative to C3 plants. And in general, C4 plants are favored in hot and arid environments. However, C3 plants may benefit more from an atmosphere that is high in CO2.